You have, let's say we have a given allele, but we have two genes for the same thing. Depending upon the characteristic, one or both genes may be expressed. Sometimes, you think of the ADO blood group, for example, sometimes there are more than two alleles for a given gene. So I want you to be thinking about dosage, sort of gene dosage response. The idea here that the number of available sequences of DNA are going to influence the amount of RNA product or protein product. All right. Now, in most cases, it's going to be either the male or female allele that's expressed, as I don't have to go sometimes neither, but that's the general rule. Now, human females present a bit of a problem because you have homologs that are identical. All right, you have human females have two X chromosomes. So in theory, for example, what's the problem? We talked about in a karyotype. One of the things you may be able to detect is something called trisomy. You may have too many chromosomes for 21, for example. Why is that a problem? Why is having too many chromosomes an issue? Why not have 4 or 5?
Overexpression of some gene.
Overexpression, exactly right. So the more of that DNA that you have for any given gene, the greater the likelihood of overexpression. And you can look at something like any of these polyploid diseases. That's not quite the correct term would be aneuploid diseases. And you see that overexpression has adverse consequences. So my question for you is, why aren't human female genetic monstrosities for that reason? Because I'm not saying they aren't, but why wouldn't overexpression, where you have identical homologs, potentially dual expression?
X inactivation of one of the chromosomes.
So what do you mean by X inactivation?
The repetitive genes get inactivated on one of the chromosomes. Only one of the two X's are expressed.
All right, that's exactly right. So I want you to be familiar with lionization or X inactivation. And all we mean by that is that the genes, typically about 85% of the genes, and that's another problem, but about 85% of the genes on one of the X chromosomes, remember you got one from mommy, the female's going to get one from mommy and one from daddy, are going to be turned off. So you know the next one. Is it the mommy chromosome or the daddy chromosome that gets turned on?
Depends.
And which would you prefer? And as it turns out, it's purely random. And it varies from tissue to tissue. Not only does it vary from tissue to tissue, I was looking for some slides I have. We'll use those in a later course. You can look at within the same tissue. Look at the brain, for example, and adjacent cells will express one X chromosome. The cell right beside it will express the daddy chromosome, mommy in one, daddy in the other. So you should appreciate that X inactivation or lionization is random, that it only applies to about 85% of the genes, so that there is still going to be overexpression in females. Why it isn't a problem, I can't tell you. I'm not sure that's fully understood. And even more bizarre, triple X females are usually fairly normal. But with each additional X chromosome, because of the overexpression, there tend to be increased issues. So we generally call individuals like that poly-X chromosomes, poly-X individuals. And the more of the X chromosomes, the more the issues. Now, it is worth noting that the process of X inactivation is achieved apparently by a glob of RNA that sort of smothers one of the X chromosomes, again in this sort of random fashion. Now, can you think of any potential therapeutic benefit if that, it's called the XISP gene, codes for that RNA, if that could be trained or directed therapeutically in some way? Can you think of any potential benefits? Any disorders involving overexpression that might be addressed by such a technique? Try some. Suppose you could turn off one of those extra 20 chromosome 20. It's been done in animals. So perhaps something to look forward to. By the way, that inactivated X chromosome, or that inactivated X chromosome often turns up what's called a bar body. You sometimes see it in neutrophils and blood stains. You sort of see one in a neutrophil here. Bar body. All right, so we're looking at the top here, what's called an indifferent or bipotential human, generally up to about 40 days or so. Humans are bipotential. And by that we mean they have really both male and female plumbing. They have a little bit of both, and they can go either way at that point. And there are two little plumbing systems in there. One's called the Mullerian duct, the other is called the Wolffian duct. The Mullerian duct will give rise to female structures. The Wolffian duct will give rise to male structures. So clearly what must happen through the course of differentiation after that bipotential stage is that one of these must be expressed and the other must be regressed or removed. And so when we talk about differentiation, you can see differentiation of the testes begins at about the 7th week, ovaries a little later, usually about the 12th week. What we mean is that these bipotential structures, one of them is differentiating into the mature plumbing, the other one is regressing. Now, in the illustration here, you see some of the specific structures that are present, for example, in the bipotential individual less than 40 days or so, for 6 weeks or so. For example, notice the genital tubercle. Over on the left, we have the female differentiation. On the male, we have on the right, we have the male differentiation. The genital tubercle gives rise either to the clitoris or the penis in the two sexes. But it's simply a matter of differentiation. Of course, we need to talk about what factors are responsible for that differentiation. You can see the structure here called the urethral groove will give rise to the vestibule and the labial region in the female. And in the male, what's sometimes called the raphe, the penile raphe, that little, we've talked about the raphe being a central line. But again, different structures developing from the same initial set of tissues. So let's talk a little bit about what is responsible for this differentiation. Now remember, males have an X chromosome and a Y chromosome. That Y chromosome we've indicated is fairly worthless, except at the very end of one of those short arms of the Y chromosome, there's a region that contains this gene, the SRY, the sex-determining region of the Y chromosome. And that's actually on a region of the Y chromosome, the short arm of the Y chromosome, that's said to be pseudo-autosomal, which is, I think we talked about what makes autosomes different from sex chromosomes. They can exchange genetic material. So technically, the only portion of the Y chromosome that can exchange genetic material with the X chromosome is that very tip out there. And that very tip is important because that very tip contains the gene that is necessary for male sex determination. Remember how we defined sex determination earlier. You might be asking yourself, what would happen if that SRY crossed over to the X chromosome during prophase of meiosis 1? But in any case, I want you to understand that this SRY gene is absolutely critical for normal male differentiation. And of course, we're only talking about the sex determination, at least initially. The SRY gene, it is a gene. There are no introns in that gene. Codes for a protein called testes determining factor, or TDF. So SRY codes for TDF. TDF is a transcription factor, and you know what transcription factors do? They turn genes on and off. TDF turns on a number of critical male genes that are necessary for male differentiation and development. We can look at some of them here. Mullerian inhibiting hormone. It's also called anti-Mullerian hormone or Mullerian inhibiting substance. Goes by a variety of names, but I think the name tells you what it does. What does it do? It's released by, yeah, it promotes regression of that Mullerian tubule. Regression of the Mullerian tubule, and that would be necessary for development of the Wolffian tubules into the male reproductive system. TDF is also necessary for expression of testosterone and dihydrotestosterone or DHT, both essential hormones for male differentiation. So over here on the right, by the way, you can see the approximate location of that TDF from the SRY on the Y chromosome. This Y chromosome is not going to have that. But in any case, right down here, this is a very important little paragraph I want you to think about and make sure you understand. Notice what it says there: male genes are needed to form male structures. It's about the male genes. We're primarily talking at this point about SRY coding for TDF. And the TDF turns on genes in other chromosomes, including the X chromosome, chromosome 20, and various other chromosomes that code for some factors that are necessary for male differentiation. So the male genes are needed to form male structures. Male structures, in particular the testes, are needed to make male hormones. All right? Testes are needed to make male hormones. And then the male hormones are needed to complete male structures. So you get a vicious regression here. You've got to have the genes to make the structures, to make the hormones, which make the structures. And, right? And if you don't have that loop in place, there's going to be a problem, at least in the differentiation of the male. And the other part of this story is if there is a problem in the differentiation of male, what is the phenotype? We have to be careful with our terms here. What is the likely phenotype if, well, let's say there's no SRY. What's the phenotype? Male or female? Female. If there's SRY but no TDF, what's the phenotype? Female. If there's SRY and TDF, but there's insufficient testosterone or testosterone receptor or insufficient MIH or MIH receptor, what's going to be the phenotype? Female. So there are a lot of ways that a female can arise. It's a lot harder to get a male, at least in terms of the biology. We can talk about that a little later too if we have time. Might be asking yourself, are there more males conceived or females based upon our discussion to this point? So there's a little sequence over here. We've talked about SRY, TDF, the importance of these male genes, and then we're going to talk about some of these critical cells in the testes. But here's the important thing: without SRY, there's going to be female development, and that's not the only way that female development rises. Female is generally the default. And if anything goes wrong in that cascade of factors leading to male development, to one degree or another, the individual will have a female phenotype. So we've talked about determined sex. Determined sex we've indicated is pretty much genetic sex, XX or XY. Of course, those are not the only alternatives. Hopefully now you realize that the individual could be XXX. The individual could be X, just one X, Turner syndrome. There was a Ryan Reynolds movie about an XXY, which he was XXY. Klinefelter syndrome. There's a whole range of these. So when we talk about genetic sex, we're just basically referring to the complement of chromosomes. Again, gonadal sex, we've indicated, generally refers to the relative preponderance of plumbing. Are the female gonads or the male gonads present? Again, not really addressing whether they're functional. And then external sex is another term for phenotypic sex. And so all that, by that we mean what does the individual appear to be? Hopefully by now you realize that there is no necessary biological or even logical consistency between these three. Right? For example, the individuals in the picture here on the right, are they male or female? And how do you make that determination? What's their phenotypic sex? Female. In fact, they're all XY. All right, so let's figure out why. These are all genetically male, so these are the determined sex. They have SRY, they had TDF. They had enough TDF to make testosterone. They had adequate amounts of DHT. They had anti-Müllerian hormone. So what was the problem here? How can they have all of these things and still have what appears to be a female phenotype? And the answer is they have something called androgen insensitivity syndrome. It's analogous to type 2 diabetes. They have the testosterone, they have all those essential characteristics, but the body does not respond to the testosterone because there's a deficiency or dysfunction of the testosterone receptor. So yet another way in which the female phenotype is sort of the default. Do we all understand in this succession of, yeah?
I'm just curious, would they be aware that they are genetically XY?
You're the PA, I'm going to ask you that question. When are they likely to become aware of that? Yeah. Or the parent might bring the child in because something isn't exactly right here. It's often the case that this is not discovered until puberty or even later in some cases. And my point is, you're the one that's going to be dealing with this. This is an abstraction for me, something that you are going to see this. All of these things we're talking about are fairly rare. But when I say rare, it's not that rare. This is sometimes called pseudo-hermaphroditism. And that term is no longer used, politically correct, and there's some other terms that are used. But what is a true hermaphrodite? Yeah, so the individual has both male and female plumbing present. Usually it's bilateral, but there are various manifestations. This is called pseudo-hermaphroditism. This would be, for example, these would be called male pseudo-hermaphrodites because in AIS, it gets very complicated because it depends on how long and what the degree of receptor insensitivity and so on is. But they have male plumbing. And so they have male gonadal sex, I'm not saying it's functional, male gonadal sex, but they have a female appearance. They don't have viable functional female reproductive structures. So that's why we call them pseudo. Anybody familiar with the current terminology describing, before I go into that point, so a true hermaphroditism is very, very, very rare, on the order of ones close to several 100,000. There are many ways in which this can occur. I want you to be thinking about that. You should be able to give me right now a list of at least 6 or 7 different ways in which this phenotype would result in an XY individual. Can you do that? Can you think of some other ways this could happen? What about if the individual has SRY but not TDF? We talked about this before. Suppose they have SRY TDF, but they don't have the gene for AMH. Or they have the gene for AMH, but they don't have receptors for AMH. Suppose remember I talked about taking that SRY gene during prophase of chromosome or meiosis 1. Suppose it crosses over the X chromosome. What do you have then? Luis was afraid of getting expressed, so he's moved to the back of the classroom. It happens. I've seen it before. You would have a normal XX male. A normal XX male. You see what I'm saying? If that SRY moves over to the X, again, that Y chromosome doesn't do much else anyway. So now you have the SRY over on the other side. That's another way it can happen. So I just want you to be aware that this is not that uncommon. Usually on the order of, collectively, all of these things are probably more common than, I know it's much more common than 1 in 1,000. Probably talking about 1 in several hundred collectively. But again, there are many ways that this can happen, and there are different degrees of it. So I was asking about the terminology. What would be anybody familiar with the terminology? Because it's been in the news with all of this discussion of what's been in the news. I hesitate to even get into the topic without get fired or something. But have you heard the term intersex? Yeah, you will. The other term that's often used to describe all of these conditions, I'll give this is male pseudohermaphroditism. Again, that's the antiquated terminology. It's also called Swyer syndrome. Swyer syndrome. So probably you should know both terms. I don't know that either are particularly helpful biologically or whatever. I think they're probably in there somewhere, but this term is sometimes used, I would argue, completely without any validity, because this also includes gender. And the other term is, you'll see, is, Swyer syndrome, to describe the male pseudohermaphroditism.
Is it possible to have female pseudohermaphroditism?
Yeah, we're going to talk about that next. But I just want, this is much more common. There's really, female is a little more difficult, and there's really, as far as I know, only one typical pathway by which that occurs, and that's less common too. This is, as I say, much more common than many of the others. All right, so Bob is asking about female pseudohermaphroditism. So this will look familiar to you. We have pituitary, we can see the hypothalamic pituitary axis, and we can see the adrenal gland there. And you know, in the adrenal cortex, there's going to be a formation of a variety of steroids, including, for example, the aldosterone, the cortisol. Remember, this is where the weak androgens are made. Now, is the production of androgens by the adrenal gland more important in the male or the female? Female. Why? Because males are going to get their androgens from where? The testes. Right? So what are we looking at here? We looked at something like this when we talked about the pregnenolone steel. In this particular case, we have the steroidogenesis going on in the female adrenal gland, the female adrenal cortex. And remember, between every one of those arrows represents an enzyme. Right? But what's shown here is that this critical enzyme, I think it's a hydroxylase 21, is missing. So as a consequence, the individual will not have adequate amounts of cortisol or glucocorticoids. That same enzyme is necessary over here to make what? Aldosterone. All right? So this individual is, and this is what I want you to understand, because of and there are many types of this particular disorder. This is the most common, or one of the most common forms. Because they're missing perhaps a gene that codes for this enzyme, they're going to have insufficient amounts of glucocorticoids and mineralocorticoids. Now, what is this dashed line again? Feedback. Now, we talked about this with the thyroid gland. This is feedback. Is the feedback going to be normal in these conditions?
No.
Is it going to be increased, elevated, depressed? How would you describe feedback back to the brain? It's going to be depressed or reduced. So that means that the pituitary is going to tell the adrenal gland to keep making more of this. But can the adrenal gland make more of this? No. What is the adrenal gland going to turn out? The only thing that it can make. The weak androgens. And developmentally, you can imagine this happening in a very young child. It could be happening even in the fetal, usually not the fetal, usually very early in the child. They're going to be cranking out copious amounts of weak DHEA, androstenedione, which are converted, we'll talk about this later, converted peripherally to testosterone. They can happen in a variety of ways. So what we have is in a genetic female, perfectly normal female, we have overproduction developmentally of androgens. And this would be, have you ever seen one of, again, I'm going to get in trouble. Have you ever seen one of the female bodybuilders that has injected themselves with androgens? Notice how their voice goes way down. Big, powerful muscles. Yeah. Imagine that happening at a very early age. So this is called congenital adrenal hyperplasia, and this is the most common type of, and you'll have no trouble going on Google or whatever and seeing some of these pictures of some of these individuals. This is the most common type of female pseudohermaphroditism in which depending upon a variety of factors, the individual is genetically female, but may have, and in often cases do, there's a famous case of a male, a weightlifter, who was genetically female, but by all accounts, by every appearance, is a very powerful male for this reason. Does everybody see the mechanism there? Overproduction of the male hormones. And there are a number of ways in which this can occur. But again, I don't like this terminology, but I think it maybe is instructive for our purposes. Does everybody know what we mean then, or do you see the distinction between a male and a female pseudohermaphrodite? Again, most of these individuals are now described as intersex or having Swyer syndrome. And there's a table here. Do you have this table? I don't know if I put it in the slides or not, but it's a table of the various possibilities here. So you can look over this. I'm not going to ask that you memorize it, but this might be helpful in trying to sort out how all of these disorders may or may not occur. In some cases, they're associated with genetic orders. In general, if there's a developmental issue, it's almost always endocrine. Can be genetic, but there's usually an endocrine problem that is directly responsible. And then in the pediatric court, she'll talk about developmental delay and precocious puberty and so on. We won't worry about that either. What do you know about foot size, shoe size? Yeah, it's false. But this is true, and if you do look at the primary literature, you'll find all sorts of NIH grants that have been devoted to this particular principle. For example, finger length and aggression, finger length and criminality, finger length and so on. Now, when we talk about finger length, what are we talking about? Am I familiar with this? Now, from now on, all the women are going to find the guys when they're going to be walking like this. Hey, how you doing? Because we're looking at the ratio and the length between the index finger and the ring finger. Index finger and the ring finger. And it's been explained in a variety of ways, but in general, what tends to happen in the male, as you can see, is the normal genetic male should have a shorter index finger than ring finger. And that is the most evident, what's the word I'm looking for, phenotypic correlate of male virility. And of course, there's good and bad to that. It probably means you're more aggressive. It probably means you're more likely to end up in San Quentin or whatever. All right, so let's talk a little bit about meiosis. That's a little bit broad.
Meiosis is the...
All right, so I'm going to talk a little bit about the formation of the gametes. Any questions then about the basic genetics? Just be aware for those developmental stages, be aware of the conundrum that you have to deal with. And this is something that you, because again, the patient who comes to you, the parent who comes to you with their child isn't really concerned about your personal opinions on the topic. And that's what most astounds and amazes me when I talk with some of the medical professionals I've talked with. Not only the ignorance, but the underlying belief system can sometimes. Because again, the patient's not concerned, the parents aren't concerned, they're just coming to you for help and for explanation. And that's why I think you need to be aware of some of these components. All right. So now we talked about gametogenesis. And so gametogenesis in humans occurs, in mammals occurs through a process called meiosis. So let's work through a little bit through meiosis. This one's a little bit tedious, but this is a useful introduction.
The process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring.
There are a few steps in between.
Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of two identical strands of DNA. Meiosis involves two divisions. The first division, meiosis I, halves the number of chromosomes, and in the second division, meiosis II, the sister chromatids are split. The end result is 4 haploid cells.
In the main.
Now let's look at each of these divisions in more detail. Meiosis I begins with prophase I. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, two homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase I, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase I, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase I, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis I, which progresses from prophase I to telophase I, two cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis II, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis I. In prophase II, we now have two cells, each with two chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase II, the chromosomes line up along the equator. This is different from metaphase I, where the homologous chromosomes were lined up. In metaphase II, each cell has only one of each homologous chromosome. In anaphase II, the sister chromatids move away from each other along the spindle fiber, and in telophase II, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
So there are several points before I come back. There's another animation here, but I want to make sure we understand some central points here. So meiosis is necessary in gametogenesis. So reproductive or species that reproduce sexually that undergo the process of making gametes, in this case either the sperm or the egg, do so through the process of reductive division or double reductive division called meiosis. And what you need to remember from your basic biology is that before any type of cell division can occur, and by the way, meiosis technically refers to division of the nucleus, cytokinesis is division of the cell, when you talk about cytokinesis there. But before any cell division can occur, be it mitosis or meiosis, you need to appreciate that the DNA must replicate. The DNA must replicate before this whole process kicks off. And remember at the beginning of this little animation, that happened during interphase. All right. Now, one of the curious things about this type of cell, it originates with a dedicated, again, the term term we've used in the past is oligopotent or multipotent stem cell. Those stem cells that give rise to sperm or eggs are diploid, and they can do one of two things, and this is important. We'll talk about this later. They can make more of themselves. So let's be sure we understand what we're saying. These stem cells that give rise to sperm or eggs can make more of themselves. What's that process called if they make more of themselves? Mitosis. So they can undergo mitosis, making more of themselves, and I'm simplifying this. Or they can begin the process of differentiating themselves into a sperm or an egg. And we just call that differentiation. But once they do that, there's no going back. And that is the beginning of meiosis. So just make sure you understand those we're going to call them spermatogonia or oogonia. The gonia at the end indicates that's the special stem cell that gives rise to either the sperm or the egg. And it can go either way. It can either make more. And of course, it has to be both. In reality, we won't get into the details of that. But it has to make more of itself from time to time, and it also has to begin the process of differentiation to become what we're going to call the primary oocyte or the primary spermatocyte. All right, so that's very important. In that animation, you saw there were 4 reproductive cells resulting from meiosis. That only happens in the male. So male, you get 4 spermatids from one stem cell. Female, you only get 1 egg. We're going to call it an oocyte. We'll quibble with the terminology later. 1 ovum, or whatever term we'll use, we'll be more careful, from the oogonia. So that's another important difference. Now, the little video here, little animation, points out some of the differences between spermatogenesis and oogenesis.
Let's compare spermatogenesis with oogenesis. Both processes start before birth, when embryonic germ cells differentiate into spermatogonia in the testis of a male, or oogonia in the ovary of a female.
Oops.
Both types of cells are diploid. However, whereas
I guess these animations I can't pause. So very important point there. Both these processes begin in gestation. Both the process, the stem cell formation begins in gestation in both. And notice the terms again: male spermatogonium, that's the stem cell. Oogonium is the singular for the female stem cell. And now I can't get it to do that because the button is so tiny.
Let's compare spermatogenesis
I've learned my
with
lesson.
oogenesis. Both processes start before birth, when embryonic germ cells differentiate into spermatogonia in the testis of a male, or oogonia in the ovary of a female. Both types of cells are diploid. However, whereas mitotic divisions continue to generate new spermatogonia in the male until death, in the female, the generation of more oogonia by mitosis halts well before birth. Although spermatogonia develop into primary spermatocytes throughout life in the male, in the female, some oogonia develop into primary oocytes, but only before birth. Males can continue to produce viable sperm from puberty until death, but females can produce viable eggs only from puberty until the supply of primary oocytes is depleted. Meiosis I does not begin until puberty in the testis of the male. Meiosis I begins before birth and ends at puberty in the ovary of the female. Meiosis II occurs within the testis at any time from puberty until death in the male. Meiosis II begins within the ovary only at ovulation in the female and ends within the oviduct upon fertilization by a sperm cell. The final result of spermatogenesis is 4 haploid sperm cells for each spermatogonium. In contrast, oogenesis can result in 1 egg cell and 2 polar bodies for each oogonium, but only if fertilization has occurred.
All right, so let's talk about some of these critical differences. I didn't have the manual dexterity to stop it. First of all, make sure, as we indicated before, that this process of differentiation begins in gestation, before birth, in both cases. However, the remarkable thing, I suppose, about the female spermatogenesis, or I'm sorry, female oogenesis, is that all of the stem cells, all of those oogonia are formed before birth in the female, and in fact, they reach their maximum number, usually a few million. And in gestation, that number begins to decline. So already in gestation in the female, the number of stem cells begins to decline. Furthermore, meiosis 1 begins in gestation in the female and continues to puberty in the female. All the while, the total number of egg cells, that we're going to call them egg cells, the number of oogonia decreasing. Then, with each ovulatory cycle, meiosis 2 in the female begins. But meiosis 2 is only completed with fertilization. So it's theoretically possible for meiosis 2 never to be completed in the female. Now the male, different story. Male, the number of stem cells can continue throughout this whole process. Generally kicks off, cranks up around puberty, and from puberty to death, whether you like it or not, the male is able to crank out both spermatogonia and spermatids. In other words, spermatogenesis continues for the entire lifespan. We just recently celebrated the 4th. Aaron Burr, for example, the famous fellow that shot Alexander Hamilton in a duel. He supposedly had children into his 80s. It's possible. But very, very important distinction. And then at the very bottom you see the spermatogenesis in the male is going to result in 4 haploid sperm cells, in the female only in 1 haploid egg cell. I'm a little careful about that. I'm reluctant to use the word egg cell there, but that's all right. So in that animation, remember he said, and so let's just work through this. I use the finger drill. You can use whatever you like to illustrate this principle, but remember all the chromosomes in these zygotes are going to involve mommy and daddy components. There's going to be a male homologue or mommy homologue and a female homologue. Now, that's going to give us our sort of traditional X chromosome. But what has to happen before we bring these together, before I begin the process of meiosis, what always has to happen for any type of cell division? You have to replicate the DNA. So I'm going to begin like this. All right. So I have the male, or let's just say the gamete chromosome, either from the male or the female. And before I can begin the process of meiosis, that has to be replicated. So I begin like this, mommy and daddy. Right? Now, meiosis consists of 2 divisions. The first division is separation of you tell me, do I separate these 2 or do I separate mommy from daddy? Mommy from daddy. All right, so the first division is separation of the homologs. These are the homologs, but notice they're still paired. We created the pairs when we did what? When we replicated the DNA. So these are the chromatids. These are sometimes called the sister chromatids. So in meiosis 1, you separate the paired homologs, and then in meiosis 2, the chromatids, the sister chromatids, are separated. So all dependent upon the succession of divisions. Now, in that process, when I began like this, and we separate the homologs first and separate the chromatids, at what point do we go from diploid to haploid? He mentioned it in the animation. Anybody notice that? Where do we go from the first division or the second division? Second. A lot of people saying second. Of course, as it would turn out, it's the first. So we go from diploid to haploid because remember these are genetically identical. So you remove the genetic contribution from mommy and daddy when you separated them in the first division. So The transition from diploid to haploid occurs in meiosis I.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs.
So you can do that
In this
calculation.
hypothetical cell with 3 chromosome pairs, 8 2 to the third possible orientations exist. Each orientation
Or 23
produces
chromosomes.
gametes with different combinations of parental chromosomes.
So the question becomes, why are you so different from your siblings, or why are you so different from the person sitting beside you? You should be able to name several reasons for the variation in your phenotype relative to everyone else. This is one cause, because of the independent assortment of the chromosomes during meiosis I. Also happens in meiosis II as random differentiation, random separation. Although curiously, neither of those is the primary cause of the reason that you look different than the person sitting beside you. You might want to ponder that. What would happen if we saw the breaking apart of the chromosome pairs? What would happen if that didn't occur? What disorder might it result? Trisomy through non-disjunction, or nullisomy, which would be the monosomy Turner syndrome. So what you should know is that if you have an abnormal number of chromosomes, it's almost always the consequence of a non-disjunction event in meiosis I or II. So here's a general summary of the most important differences between male and female gametogenesis. Remember, female, all the oocytes are present at birth. The male keeps cranking them out throughout life. In the female, each gamete is released with each ovarian cycle. We'll talk about that next time. And then remember, meiosis I in the female begins in gestation, but and goes until, or continues until each pubertal ovulatory cycle. Meiosis II begins with each of the ovulatory cycles and is only completed with fertilization. Once again, the male differs because the male can just keep cranking these out from puberty to death. The other important distinction is that we pointed out in the female, there's only generation of one gamete per meiotic event. In the male, you get 4 spermatids from each meiosis. In the female, with the first division, you get cytoplasm and you get something called a polar body. Polar body decays. In the second division, you get cytoplasm and another polar body, and that polar body also decays, such that at the end, and by the way, don't confuse the polar body with the bar body. By the end, you only have one functional gamete in the female. All the others are polar bodies. All right, we'll stop there. And so I guess that's as far as we'll get for your test. Actually, so your test is Wednesday this time, isn't it? Give me a moment to consult with myself. Yeah, we'll be able to finish this. So there are only a couple slides left, so we'll finish this. Monday, I guess. Yeah, I'll make a decision. That'll be an executive level decision because we're going to have to borrow one of those. I'll let you know. So again, your first test is on this material.